Good morning everyone. In the last module, that is vacuole 1, we have studied the structure of vacuole, plant vacuole, the types of plant vacuoles and biogenesis of plant vacuole. In this module, that is plant vacuole 2, we are going to study the functions of plant vacuoles. As we know, vacuoles are membrane bound organelles and it plays important role in plant cell system. It is multifunctional organelle which helps in to maintain cell size, cell shape and to control turgor pressure, also helps in storage, that is temporary storage, and plays important role in membrane transport. So, learning objectives of this module are functions of vacuoles in that transport across tonoplast, autophagic activity of provacuoles, transport on the tonoplast of plant cells, protein transferred to the vacuole, sorting of proteins from the late Golgi to the vacuole. Functions of vacuoles. Vacuoles are membrane bound structures within the cytoplasm of a cell that function in several different ways. In mature plant cells, vacuoles tend to be very large and are extremely important in providing structural support as well as serving functions such as storage, waste disposal, protection and growth. Many plant cells have a large single central vacuole that typically takes up most of the volume in the cell, around 80% or more. Vacuoles are commonly used to temporarily store material or to transport substances. The central vacuole stores salt, minerals, nutrients, proteins, pigments, helps in plant growth and plays important structural role for the plant. Among its role, plant cell function, the central vacuoles, as we know, stores salt, mineral, nutrients, protein, pigments and helps in plant growth and plays important structural role for the plant. Under optimal conditions, the vacuoles are filled with water to the point that they exert a significant pressure against the cell wall. This helps to maintain the structural integrity of the plant cell. Along with the support from the cell wall and enables the plant cell to grow much larger without having to synthesize new cytoplasm. In most cases, the cytoplasm is confined to a thin layer positioned between the plasma membrane and the tonoplast, yielding a large ratio of membrane surface to cytoplasm. Under optimal condition, the vacuoles are filled with water to the point that they exert a significant pressure against the cell wall. Turgor pressure helps to maintain the structural integrity of the plant cell. The structural importance of the plant vacuole is related to its ability to control turgor pressure. Turgor pressure detects the rigidity of the cell and is associated with the difference between the osmotic pressure inside and outside of the cell. Osmotic pressure is the pressure required to prevent fluid diffusing through a semi-permeable membrane separating two solutions containing different concentrations of solute molecules. The response of plant cells to water is a prime example of the significance of turgor pressure. When a plant receives adequate amounts of water, the central vacuoles of its cells swells as the liquid collects within them, creating a high level of turgor pressure. This helps to maintain the structural integrity of the plant along with the support from the cell wall. In the absence of enough water, however, central vacuoles shrink and turgor pressure is reduced, compromising the plant's rigidity so that the wilting takes place. Plant vacuoles are also important for their role in molecular degradation and storage. Sometimes these functions are carried out by different vacuoles in the same cell, one serving as a compartment for breaking down materials. Transport across tonoplast. The vacuolar membrane or tonoplast is a selective membrane and the passage for chemicals through it is controlled in both directions. Water can pass in and out freely, but other small molecules are retained within the vacuoles. Many proteins arriving at the cytoplasmic surface of the vacuoles are synthesized by membrane bound ribosomes of the rough endoplasmic reticulum and transported to the vacuole via the Golgi apparatus. Part of the vacuolar membrane works as a proton pump and use energy from adenosine triphosphate, that is ATP, to pump hydrogen ions into the contents of the vacuole. These maintain acid condition inside it. A working model for transport pathways in vacuolar apparatus. A working model for transport pathways in the vacuolar apparatus. It has been put forward by Marty in 1999. He has suggested that 7 basic pathways are used for the biogenesis, maintenance and supplying of vacuoles. These pathways are: pathway 1, where entry and transport in the early secretory pathway, that is from ER to the late Golgi compartments. Pathway 2, where sorting of vacuolar proteins in the trans-Golgi network, that is TGN, to pre or pro-vacuolar compartment PVC via an early biosynthesis vacuolar pathway. Pathway 3, where transport from PVC to vacuole via the late biosynthetic vacuolar pathway. Pathway 4, where transport from early secretory steps, that is ER to Golgi complex, pathway 1, to the vacuole via alternative route with possible material accretion from Golgi indicated by the asterisk. Pathway 5, endocytic pathways from the cell surface to the vacuole via endosomes. Pathway 6, cytoplasm to vacuole through autophagy by degradative or biosynthetic pathways. Pathway 7, transport of ions and solute across the tonoplast. These pathways are depicted in the figure and Short forms used for these figures we have given in the figure itself. In this figure we have studied sequential stages of cytoplasmic confinement by provacuoles involved in cellular autophagy have been reported by Marty in 1999. Tubular provacuoles 1 and 2 form cage-like traps 2 to 4 enclosing portions of cytoplasm of a cell from the root meristem of euphorbia. Adjacent bars of the cages then fuse to build a continuous and tight envelope, which is central structure 4, around segregated portions of cytoplasm. The specimen, that is 3 mm in thickness, was examined without counterstain at 2.5 MV with a very high voltage 3 MV electron microscope. Provacuoles are shown in yellow color, Golgi complexes in red color, and mitochondria and plastids in blue in color in the following figure. Model of ABC transporters, H plus primary pumps, H plus coupled transporters and channel in a simplified tonoplast. Glutathione S conjugate, that is GSX, and metabolite, that is M, transport is achieved by an ABC transporters. In that, tonoplast shows presence of various mechanisms of transport. A model of ABC transporters, H plus primary pumps, H coupled transporters and channel in a simplified tonoplast has been proposed by Marty in 1999. It is suggested that glutathione S conjugate, that is GSX, and metabolite M transport is achieved by ABC transporters, and electrogenic, that is H plus ATPase V type and H plus prophosphatase PPases are involved in acidification of vacuoles. The proton motives force provides energy for uptake and release of different solutes, that is cations, anions and organic solutes. Donated A, B and C indiscriminately in the figure across the tonoplast through transporters and channels. Water channels, that is aquaporins, facilitate the passive exchange of water across the tonoplast, which also leads to passive transport of small ions and molecules. Transport system of the tonoplast, vacuolar membrane of plant cells. In this, two electrogenic proton pumps driven by ATP and phosphatases, that is PP, generate the APH and A4 asterisk for the secondary transport of other solute into the vacuole. SV, that is slow vacuolar channels, FA, that is fast vacuolar channels. Scheme of mechanism and route of protein transfer to the vacuole. Upper part, that is vacuole generation during cell division by de novo generation or appropriament from mother cell. Lower part, that is transport of protein into the vacuole. Proteins can be arrived by endomembrane C. Progression when they are transported by vesicular transfer via Golgi apparatus, which is given by different authors, that is Nishimura et al. 2004, Herman and Schmidt 2004, etc. Vesicular transport of proteins from the endoplasmic reticulum to the Golgi is indicated by a white circle. On their way from the TGN to the vacuole, many proteins with the sorting determinants are contained in transfer vesicles and such as DV or CCV, which discharge their cargo aid to MVB or PVC, or might even bypass this endosomal compartment. The ER, that is endoplasmic reticulum, generates several vesicles, that is PAC, ER bodies and PB, where proteins can be transport to a vacuole without progression through the Golgi. A specific variant of this mechanism represents the direct formation of PB from the un-ER. Autophagy transfer cytoplasmic proteins or organelles into vacuoles. PM proteins can be transported to the vacuole by endocytosis. CCV, that is Catherine coated vesicle, then CTVSD, again C-terminal vacuolar sorting determinant, DV, a dense vesicles, ER, endoplasmic reticular, and LV, that is lytic vacuoles, MVB, that is RSI multivesicular bodies, PB, that is protein bodies, PM, plasma membrane, PSV, that is protein storage vacuole, PVCs are pre-vacuole compartment, and PAC is nothing but precursor T, accumulating vesicles. Now, SSVSD is the sequence specific vesicular sorting determinant, and in this figure Greek letter indicates characteristic isoforms of TIPs of different types of CH of vacuoles. Schematic overview of the functions of vacuolar proteolysis in cellular protein breakdown and amino acid recycling. Vacuolar proteolysis, that is nothing but lytic vacuoles. Functions of vacuolar proteolysis in cellular protein breakdown and amino acid recycling. Precursors of vacuolar proteases are synthesized at the endoplasmic reticulum and transferred in vesicle to the vacuoles either via endomembrane progression through the Golgi and the MVB, or bypassing the Golgi, that is packed precursor protease vesicles and ER bodies, ribosomes, etc. Okay, lower part in MVB and vacuoles, that is LV, PSV, precursors of processing enzyme, example VPE, are activated autocatalytically by limited proteolysis precursor processing. Activated processing proteases, other precursors polypeptides and thereby active enzymes and generate polypeptide involves in defense processes. Proteins to be degraded get into vacuoles. Only vacuolar proteins transferred by variant of autophagy is indicated here because of its specific contribution to senescence and PCD. Limited and complete vacuolar proteolysis is a major process in cellular amino acid recycling after a long term storage of protein reserves, when selected proteins are broken down immediately after getting into the vacuole or during senescence. Mega autophagy represents the final step in PCD when the release of proteases leads to cell corpse degradation. sorting of proteins from the late Golgi to the vacuoles. Vacuolar proteins are sorted in vesicles bound for the vacuole at the level of TGN. The soluble hydrolases CPY binds to its membrane receptor, that is VPS10P in this compartment, and the receptor ligand complex is delivered to PVC. In the PVC, CPY dissociates from its receptor and travels on the vacuole, while VPS10P returns to the Golgi to bind more ligand. A second pathway from the TGN to the vacuole does not pass through this compartment and is taken by the vacuolar membrane proteins ALP and VAM3P. Interestingly, the two pathways converge at or before the vacuolar membrane, and this is depicted in figures. Research in intracellular trafficking in plants is gaining momentum. Markers for different pathways and compartments of the endomembrane system are now available, and genetic dissection of biosynthetic, endocytic and autophagic pathways is underway. This will lead to more insight into the transport across vacuolar membranes. These are the references. To summarize this module, as we know, vacuoles are important multifunctional organelles of plant cell system and which plays important role in storage, especially central vacuole, which store salt, minerals, pigments as well as secondary metabolites. Vacuoles also help in maintain the turgor pressure of the plant cell. Tonoplast, that is vacuolar membrane, act as a selective membrane and controls transport across the membrane. We have also studied autophagic activity of lytic cell, that is lytic vacuole cell as well as provacuole cells. In this module, we have studied a transport model, that is ABC transport channel, and we study the transport of membrane transport of vacuoles.